Dear student, we will continue with mutation and we are going to study human genetic disorder. Now, human genetic disorders are categorized into two different categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are those which follow the Mendelian law of inheritance. They are divided into two categories: recessively inherited disease and dominantly inherited disease. Dominantly inherited disease such as Huntington's disease. Recessively inherited disease again divided into two categories: one which are present or related to autosomes and one which are related to sex chromosome. Autosomal are phenylketonuria, albinism, sickle cell anemia and thalassemia. We have already done few out of these. Sex-linked are color blindness and hemophilia. The sex-linked disorder are also called as X-linked recessive disorders. So first we will study autosomal recessive disorder. Now the defective gene are present on the autosomes in these cases. Therefore, the female and male, they have both equal chances to be affected. The disease is expressed in the homozygous recessive condition. The heterozygous are the carrier and have tendency to transmit the disease to the next generation, but heterozygous will not express the disease. So this is what is called as Mendelian pattern of inheritance. We will study here thalassemia first. So thalassemia, I'm doing one example from each so that you can understand how to study. Now thalassemia, what is thalassemia? It is so called as autosomal recessive disorder in which the affected person will have less hemoglobin than normal and you can say fewer red blood cells than normal. Thalassemia may result into severe anemia. Now what is the genetics of the disease? Hemoglobin, has 4 globin chains, 2 alpha and 2 beta. Even the thalassemia is divided into two categories. Alpha, if defect is in the alpha chain, it is called alpha thalassemia. If it is in the beta globin chain, then it is called beta thalassemia. Here I am discussing in general what is thalassemia. So mutation in the gene coding for either alpha or beta chain of hemoglobin will result in the production of defective hemoglobin. So what is the function of hemoglobin? It is the carrier of oxygen, and if hemoglobin is defective, then there is defect in the whole process of carrying the oxygen even in the RBC. So common symptoms that you see in the thalassemia patient: bone deformities, especially in the faces, dark urine, delayed growth, excessive tiredness and fatigue, yellow and pale skin. Now here I am in general discussing the inheritance pattern and you can try with other diseases. Okay, autosomes, suppose AABB. Here I am representing A is the autosome of one particular parent, maybe male or female, it does not matter over here, and because the disease is autosomal. And B, let's say, is the autosome of another parent, let's say the female parent. N represent normal condition of a person, and D represent a disease, that is mutation in a particular gene. So when we are expressing thalassemia, we can write T instead of D. If you are representing some other disease, similarly you can change. So look at the parents, male and female. So here one of the parent carry the disease gene. Both the parent they have one copy of disease gene. They are carrier because it is present in the heterozygous condition. Both are heterozygous. So when they are going to form the gametes they'll form, and the fusion of gametes occur. Let's look at the possibility. So you study the gametes. Now this will be normal. Okay, this will be again heterozygous condition, so it will the phenotype will be normal, but actually if you look at genotype, it is carrier. again here one disease gene is there, so the phenotype is normal but it will carry the disease. Now carrier have a tendency to transmit the disease to the next generation as compared to normal. And here you see both the disease, so it is homozygous condition and the disease get expressed in the homozygous condition. So this is expressed. Now if you analyze the whole cross, you will find there is 25 out of 4 there is one possibility of disease. So 25% possibility of disease individual, and you can say 50 of the carrier and 25% of the normal. Now we'll take another disease from the sex-linked or X-linked recessive disorder. This is hemophilia, which is also known as bleeder's disease. It is X-linked recessive disorder in which the blood does not form clot. The genes associated with this condition is located on X chromosome. Let's see what is the genetics. There is a mutation in the gene that makes clotting factor. So defective protein is synthesized and this protein will not contribute for the clot formation. So the person who is suffering from hemophilia, the common problem is that there is no clot. If any bleeding occurs, there is no clot formation that takes place in the body, and there is excessive loss of blood takes place from the body. Now what is the pattern of inheritance? This is very, very important. Male single allele produce the disease in male because male have X and Y. So if the disease is present on the X, it is going to express in the male. In female, 2 allele will produce the disease. So it is expressed in the female only in the homozygous recessive condition. But if the female is heterozygous, then it will not produce the disease, but it is a carrier. So carrier female transmit the disease, transmit the gene 50% of her sons. Hemophilic female will have hemophilic father. This disease is expressed in male, but it is transmitted by female. So males are never carrier. Let's understand this cross. A cross between here I have to take X and Y because this disease is present on the we can say sex chromosome. So we have to consider here X and Y. So XXH over here represent hemophilic. Male is normal. Okay. So if the female is diseased, let's talk about what is the percentage of disease to be transmitted to next generation if it is crossed with normal. So gametes are there, and then you can look at the inheritance pattern. So overall, if you look at the males, total disease production in the next generation will be 25%, and then you can say 50% are normal, 25% are carrier. And if you just look at the one possibility of normal male, one possibility of normal diseased male. So out of male there is 50% of disease to be transmitted if the child is male, and 50% possibility of carrier if the individual is carrier. Okay, so this is how you have to analyze the inheritance. Now let's see we'll continue in the next part 3. Thank you.